Hello students, welcome to this virus chapter. Virus is one of the non-living living creature. They are non-cellular, microscopic infectious agent that can only replicate inside a host cell. From a biological perspective, virus cannot be classified either as a living organism or non-living. A virus can be infectious agent which only replicate with a host organism. This is due to the fact that they possess a certain defining character features of living organism and non-living entities. In a nutshell, a virus is a non-cellular infectious entity made up of a genetic material and a protein, and they can invade, reproduce, and cause diseases in plants, bacteria, and animals. For instance, a virus which cannot replicate itself outside the host cell, this is mainly because a virus lacks the required cellular machinery, therefore it enters and attaches itself to the specific host, injects its genetic material, and reproduces by using host genome. Finally, the host cell, it can split open or else lifetime it can stay with the host and it can continue its generation. In most of the cases, they kill the host and its new particles gets released, they will go on infect. Viruses can also be crystallized, which as no other living organism can do, and it is this factor that leads to virus being classified in the gray area between living and non-living. Let us look at a little more about virus. Viruses are fascinating and enigmatic entities that have been subjected of scientific curiosity and concern since their discovery. Humans know this virus from longer duration. One important reason why humans are worried about this virus, most of the pandemics are caused by this virus. Sometimes, even though we blame virus, bacteria are also responsible many pandemics. But most of these bacterial diseases were epidemics, but these viral diseases were pandemic because their infectious rate was high. These microscopic infectious agents are responsible for a wide range of diseases if you look at a globe, and we were suffered because of COVID-19 in 2020 until we are suffering because of this. And this can be seen in humans, animals, and even in plants. And we are, as a global populations, we are looking for a more food production. So for any kind of infection to a plant is a bigger concern because that can create a devastating effect on our food chains because it's a even though we call it is a globalized thing, even though our supply and demand is met every now and then, but there is a limitation for our supply. So if something went wrong, a particularly agriculturally strong country like India or maybe in Brazil or maybe in the United States, that can go and affect the entire global food supply. So this viral infection, one side it can kill human beings or animal, but at the same time it can affect our food and nutrition balance, and even it can kill our food supply. So that's why it is causing problem to humans, animals, and plants, and in some cases even for bacteria, because this bacteriophages, they are specialized virus which can really infect this bacteria. So understanding basics of this virus and viral disease is crucial for a public health, medicine, and biology. This is the important thing because unless you understand a virus, we cannot diagnose the virus. Unless you diagnose the virus, you can't treat viral diseases. So the first and foremost task in virology is understanding more about a virus. One thing, what are these viruses? What kind of class they are? What are their structures? And what is the host reservoir? That's very important thing. Virus is from which place it is coming, whether it is from monkey, whether it is from bats, whether it is from water sources, whether it is from another human beings, or is it from other sources, maybe from other continent. Even in case of COVID-19, and it has started from Henan markets in China, but it spread across the globe. Then we can call it is a Chinese virus, which has started from China, then spread to the globe. Now it has become a global virus. So it's important to understand their structure also. Unless you understand the structure of this virus, we can't design a medicine for that. So first thing is understanding the structure of virus. Once you understood the structure of virus, you can design an antibody or medicine, or maybe you can design a vaccine for that. Because today's world, the recombinant technology, our rDNA technology is helping us to develop or fast track the process of vaccine development. So we could develop a vaccine in a shorter duration. That's important thing. Just developing vaccine is not important, and but developing a effective vaccine in shorter time is important. For that also we need to understand a virus. And then this biology, because biology is a huge field, whereas virus play important role there. When it causes a disease, one thing is biology market, particularly if you look at, it's a disease diagnosis and disease treatment oriented. So biology, when you look at from a disease point of view, how we can manage a disease. But the important thing we should remember here is the viral disease, there is no proper medications or medicine. So what only way is supportive care is given. So when you give a supportive care for a particular viral disease, how to give a supportive care or what kind of supportive care should be given. So that is a one priority area in virology. Then the community health. So that is the public health or community health is important aspects, because this virus can spread anyway. So it can travel with your goods, it can travel with any of biological sources. So one day it may appear here, another day it may appear in another places. You might have seen recently, you might have heard about the Nipah viral cases in Kerala. So what really happens? Nipah virus was initially reported in Malaysia. From Malaysia, later it was reported in Bangladesh. From there, it was reported in Bangla, West Bengal of India. But later, suddenly it appeared in Kerala, and it is somewhat like a host is prevalent there. Some areas were prevalent for many viruses, not only in case of Nipah, in other cases also. There are some area where virus is prevalent, and from there it can cause one of the biggest concern for public health and community health. So because it infect one in a family, from there it can infect whole community. So it's a biggest challenge for a whole our state machinery to curb the spread of this virus. So that's the important task, because unless you understand a virus, their disease pattern, you cannot curb their growth or curb their spread. So this course, what we are designed is to provide an overview of a virus. I mean the basic overview of virus. We cannot touch every virus, but at least a general perspective about a virus, their structure, their replication mechanism, and impact on viral disease in human and animal health. Because every now and then we listen about or we hear about a viral disease, unless you understand them from a deeper point of view, you cannot contribute more in this field. So our idea is understand a virus first, then understand how they cause a disease. Once you understood that this virus causes disease like this, how to design a treatment pattern for that particular disease. Either maybe developing a vaccine, or maybe supportive care, or maybe developing antiviral drugs, or maybe developing monoclonal antivirus to treat. Because in some cases, in emergency situation, we can't look for vaccines. So in such cases, already developed cloned, antibodies, that particularly monoclonal antibodies, can be used. As a biology enthusiast, the first question which comes to our mind is, what are these viruses? Viruses are non-cellular infectious agents. Why do we call this as a non-cellular? The important thing is they cannot sustain themselves as an individual identity. They require animal host, or an animal cell, or a plant cell, or bacterial cell for their survival. They can infect them and they can survive them. But if you ask whether this virus can survive outside, yes, it can survive outside for a shorter duration, not for a longer durations. But there are cases which survive for more durations, mainly because this virus can move to a crystallized stage. Then it can survive for longer durations. So one more important thing, they cannot carry out their metabolic process on their own, so they require host. So that's why it is classified as obligate intracellular parasites. And if you look at a virus in larger class, there may be a DNA virus or RNA virus. Or animal viruses, plant viruses are there, but at the same time, two major classes, either DNA viruses or RNA viruses. Why do we call them as a DNA viruses? The virus which contain genetic material as a DNA, we call them as DNA viruses. If the virus contains genetic material as RNA, we call them as RNA viruses. So both will cause a problem in human beings, both will cause a problem in animals, both will cause a problem in plants. So what are the basic structure of this virus? I'm not talking about exactly how the COVID virus looks, how the Ebola virus looks, how the Nipah virus. We are not talking like that, but we are talking how exactly a basic viral structure will be there. The important thing is the genetic material. There will be a strong genetic material which is sufficient for that virus to make its progenies, and it codes all the genes which is required for viral survival. So it consists of a nucleic acid core, and this core consists of either DNA or RNA, as already I told, and surrounded by a protein coat. This protein coat is a strong coat, which is also called as a capsid. And this is important for a bacterial survival because this genetic material is prone for attack from outside, because this DNA or RNA can be easily attacked with DNases or RNases. So this protein capsid will protect this genetic material in an intact way. Some viruses have an additional lipid envelope derived from a host cell membrane also in some classes, not in all. The nucleic acid encodes the genetic information required for replications. Capsid protects and delivers their genetic materials. So capsid is playing important role in protection. The genetic material can be DNA or RNA, as already I told. So there is no chance that DNA are both the cases. I mean DNA and RNA together, only DNA or RNA. But there are classes of virus called as retrovirus. They can make DNA from RNA. This virus in general, they are non-cellular organism which is enclosed in a protective envelope. The presence of spike helps in attaching the virus to the host cells. There is a spike, there is a structure through which it can cling to the host cell. It is something like how a key attaches to a lock, and how that key, if it is perfect, how it can open the lock. Same thing like that. This virus attaches the cell, open the cell, and enter the cell. These viruses do not grow, neither respire nor metabolize, but they can reproduce. That is important. These viruses do not grow, neither respire nor metabolize, but they can reproduce. They are surrounded by a protein coat, that's what you call capsid, and a genetic material, DNA or RNA. They are considered both as a living and non-living things. And these viruses are inactive when they are present outside the cell. They're active inside the cell. That's why these are challenging or tricky things, because they show both living and non-living characters. These viruses cause several infections and reproduce within the host cell by using enzymes and raw materials. So the important thing is this virus uses our system, that particularly host cell system, and make their next generations. That is the important thing, because they are using our system and developing their own genetic material, their own protein code, and make their progeny so they can infect the next cells. So it is something like that. It is using our platform and generating the information what it requires. Once it develops the informations, they make their whole structure. Once the structural integrity is maintained, then it is ready to release. So release is just, it's not releasing. Once it releases, it can infect the neighboring cells. But if you ask me whether we have a system to combat this, yes. Every evolved organism has got its own system to combat or fight against this virus. But it doesn't mean that we can contain or curb the growth of virus in our human system. That's why we have vaccine and other medication system to manage this virus. As you all remember, human cells or plant cells or maybe bacterial cells, if you look at them close by, there are genetic material inside. Then outside there are cell membranes. But in case of human cell, there is no cell wall. But in case of bacteria or maybe a plant cell, there are strong cell wall. But this virus can break both. Cell wall it can break cell membrane also. But if you look at a cell wall and cell membrane is easy to break because it is simple. A fluid, it is consist of a phospholipid which are fluid in nature. If a lock fits with the key fits with a lock, it can easily open the door and enter inside the cell. So the virals, once it is entered inside the cell, it can replicate. Replication is essential for its to make its progeny. So if you keep human cell, plant cell, and bacterial cell, which can get infection fast. If you look at a virus cannot simply replicate. Virus require a host cells. So how virus can? So important thing for a virus is attaching to a cell. Once it attached to a cell, attachment happens between the chemical molecules which is found on a cell surface and the chemical molecules which is found on a viral surface, mainly glycoproteins. The glycoproteins are the one which act as a key and lock. It attaches there. Once it attaches there, then this virus can roll inside the host cells. And this receptor base is mainly carbohydrate. I told it is a glycoprotein, so it's a carbohydrate based communications. And this entry mechanism vary from virus to virus, and sometimes virus will just bind on surface, then it injects its genetic material, or sometimes virus can go inside the cell through a mechanism called as uh, endocytosis. As we all know, endocytosis is an important mechanism through which the cell can get any materials. How the cell can get a materials? It can engulf anything. So engulfing happens in the form of endosomes. So endosome engulf material will have a virus inside and surrounded by cell membrane. One thing you should remember is these viruses are highly tricky. So once they are inside the host cells, it can hijack our whole machinery. That means we have our general machineries to supply energy and make our genetic material to maintain ourselves. But this what this virus does, they hijack the machinery so that use every possible molecule in and around that infected area or infected area inside the cell, use them for their benefits. That means to build their capsid, to make their genetic material. That means every nucleotide what it uses in the synthesis process is stolen from our cell. So our cell, our poor cell provide all the material which is required for their survival. The replication process may follow that lytic cycle. Lytic cycle, in that process, what happens is the cell will lyse and things will get released. That means the progeny will get released. But in lysogenic cycle, the viral DNA integrated in host genome and remain latent until it get activated. But important thing you should remember that, so during this whole process of genetic replications, it may take up some of our gene also. It can bring some gene from outside and put it into our system, or it can join with our system and stay for longer duration until it get activated. So the replication of a virus inside a host is a highly tricky process, and through that process it can steal all the information from us, all the genetic material, protein, and it pack it and make a new viral particle and release into extracellular space. From there, it can keep on infect neighboring cells. We try to combat with them, but the amount of viral load is important. If a viral load is more, which can infect most of the cells and create a higher and high-end symptoms. But if a viral load is low, it cannot create much symptoms because the amount of virus will be low in a host. So inside, before entering inside a cell, it is easy to kill those viruses. Once if it is there inside the cell, they hijack our machinery, they start synthesizing. Once they synthesize, they start release. But during synthesis process, we have some techniques like we can target some of the enzymes and kill those viruses. That's what practiced in HIV cases and all. ATR treatment, anti-retro ART treatments, antiretroviral treatments where they use some kind of the molecules to tap or to kill these enzymes which are involved in replications, the viral enzymes which are involved in replications. If we can tackle that, so easily we can reduce a replication rate. Once you reduce a replication rate, you can reduce the growth of that virus. So replication rate is directly proportional to the growth of that virus. So by the way, we can curb that. So if it is replicated inside host cell and get released, so it can infect a new viral cell, or else we can make our neighboring cells alert. By that way, this cell won't get infected. That is another possible way to combat against a viral disease. But these are all temporary treatment. Unless we produce a strong immune molecules, we are prone for viral infections. This is the final stage in viral replications. Once they are completely formed, its genetic material is ready, its capsid is ready. Capsids are stitched by taking individual amino acids, and finally a strong capsid is ready. Now the DNA is ready, or RNA is ready. They can pack and make a new viral particle or a young virus called as virions. Once they are ready, they can release it from host cells. Lysis mechanism it can use, or budding mechanism it can use, exocytosis mechanisms. That depends on what kind of virus they are. We can't expect everything to form a lysis mechanism. But lysis is the one mechanism through which it can break open the cell and easily get released into the environment. Budding is another mechanism, and exocytosis. Exocytosis is similar to cellular vomiting, through which the cell can throw its waste out of the cell. So same mechanism, virus also can come out of the host cell. So we have seen now what are viruses, what are the structure of virus, simple viral structures, then what are the properties of virus, how do they replicate. This all thing is in a general view. But what exactly it does or what is the impact of these viral infections or viral disease, particularly human. As we our populations are crossing 8 billion, we are living close by. So if one person gets infected with virus, the chances of spreading are very high. So that's why we are worried about viral infections. Numerous viral diseases like influenza virus, HIV or AIDS virus, COVID, SARS-CoV is another biggest challenge what we face. But important thing is we developed the technology to formulate a new vaccines, new formations or new formulations through which we can manage this disease. But every now and then they throw a challenge on us because they manipulate their genetic material in faster mode. When they manipulate their genetic material or mutate faster, so it can create a new uh, the new kind of challenge for our immune system because it is a new virus or new strains. As it develops into a new strain, we need to have a better combat machineries. Otherwise, we can't manage this virus. The viral disease can vary from mild self-limiting infection to severe life-threatening conditions. So this is the one challenging thing. Sometimes in some people, viral infections shows a mild symptoms, but it is not true in all the cases. There are people who are called as immunologically compromised people. They are immunologically compromised, so these viral infections, simple viral infections can lead to a severe complication. Or else, just imagine a person who got higher viral load. So if a viral load is very high, that person also can severe symptoms. So two things are there here. One is infection. Second is our combat machinery. When both works together, it is easy to eliminate. Sometimes what happens, viral load may be low, but our system combat in a high-end way, which is not really required. In that combat way, what happens? Our own cell will kill our own cells. Self-killing will happen. But life-threatening conditions are mainly seen in cases of some virus like Nipah. Live death rate is more than 70%, approximately 70%. And monoclonal antibody treatment is only promising path for us to treat this. And one more is Ebola. The mortality rate is more than 90%. So that's the life-threatening conditions. And there are viruses which infects our brain systems like encephalitis. So we can't treat it easily. We can't access our CSF and treat it. So it is very difficult in humor. So there is a huge impact on human economy, human development, and human growth. So that's why high-end impact we can see in case of humans. So if an animal got infected virus, just imagine a virus got causing some problems in animals. Is it really a worry? One thing is humans are close to animals. Some of them are pets, some of them are reared for a meat purpose or nutrition purpose, or some are reared for a milk, other dairy products purpose. So that means a human existence is questioned if no animals and plants are there in and around us. So we need them. We utilize them, we use them, we exploit them, and it's all for a human benefit. So this is a greater concern because if a virus infects animals, from animals it can easily transmit to human beings. That is the one thing. Or else if a large scale death happens in particularly dairy industry, just imagine if a particular virus infects and kills all the cow in particular area, maybe in the United States, what will going to happen for the dairy industry? What about the milk products? What about the meat industry? So that's a challenging thing. So that's why one more thing is pets. First, like just imagine a rabies viral infections in case of uh, dogs, because these cat families and canine families are always prone for infections faster more. They'll catch the infection from there, they will start dying, and that's with their pet. And human beings also get if they bite or something through which if a virus can enter human body, they can also cause disease in us. Then one more challenge, foot and mouth disease in particularly in cow, because some of the countries economies completely depends on dairy industries or dairy products. Just imagine all the cow or maybe the cattle got infected with this kind of virus, then who will buy their products? Then avian, it's another challenging thing, particularly on chicken or poultries. So poultry is one more challenge. We can't survive without chicken nowadays. We are more, we are more of a meat eater. So in terms of meat eating, this thing, chicken is a one of the highest supplied meat compared to other meats which are routinely used across the globe. And if influenza H5N1 or avian flu, if it attacks them, so mass scale killing happens, or mass scale uh, effect we can see on poultry industries. So because this zoonotic potential of this virus is there is no boundary for them. So because they can infect in one continent, from there it can move to another continent, and it can shift our balance in food what we maintain till now. Maybe just imagine a particular amount of chicken is needed for India, and if a shortage comes, then from where we supply, if everywhere it got infected like that, we don't buy it from outside. Just what happens to rambutan market. Rambutan is a fruit. What happens to that because of a Nipah virus? People stopped buying it, and some people who were cultivated only rambutan, they could not sell it. So this is the challenging thing. This viral infections, one side it can kill human beings, second side it can infect our create a problem in our animal sector, particularly animal husbandry sector. But at the same time, the market is the big issue. Some people are completely depends on this kind of markets. If we can't maintain that, that will going to create a devastating effect on particularly emerging markets like India, because some of the farmers are completely depends on animals or maybe plants, and if that plant or animal is vital for their economical survival, if anything go wrong with that, that will going to have a high end effect on their. Next we will move on to plant viral disease. It's another important source. One is a food source, another it is the feed source. Because when we look at a plant, we always see whether plants are required. Yes, plants are vegetables and fruits which are important part of our diet, and same time these plants are serving animals also, because some of the food feeds which are made out of plant sources. And if there is a problem in the plant, a particular, just imagine hundreds of acres of tomatoes cultivated in India which got a particular disease, and every tomato plant diet is sapling stage, and can we imagine of a cost of the can a particular country like emerging market like India can manage it? It's very, very difficult. So the plant viral disease is also important thing. So you need to curb the growth of virus at the initial stage. Then only we can manage that. But if it's spread to whole field, then one thing is managing is difficult. Second thing is treating them is also difficult. And third thing is which can lead to reduction in crop yields, and economic losses can happen to agriculture, which is already in the brim of collapse. Just imagine most of the agricultural industries, particularly in the rural sector of India, they are in the brim of collapse. If you just imagine in that one, if a viral disease also spreads, that will going to have a devastating effect on their economy. So that's why that may be a human disease, or maybe animal disease, or maybe plant disease. All this going to impact a human life in one or another way. So when we before we wind up, the important thing is virus can keep on cause a problem to us. Even though it can cause a problem to us, we can manage them nicely, or manage them uh, with our present available technology, but before it spread to a larger area. So in conclusion, viruses are tiny as we understood about them. They profound influence on the health and well-being of living organisms. So their structure, their replication mechanisms are unique. One virus replicating one way. So understanding them in a structural pattern as well as their replication machinery is important to create a kind of database where we can store information. Nowadays the technology is helping us to store the information, so anyone can access and use them whenever they need, if we have a proper structure and their sequence and other things. The impact of viral disease on humans, animals, and plant underscores the critical need of our ongoing research and vigilance in public health, medicine, and agriculture. This is the important things. So it's not just understanding about a disease is important, but at the same time, how we can use this to keep eagle's eye on this spreading, particularly in public health, or maybe in developing new medicines, or maybe particularly in area like agriculture. By deepening our understanding of virus, we can better prepare for a future problems or future infections. We can combat them and mitigate the effect of viral disease, ultimately safeguarding the health and prosperity of the world. So now we have superbugs, which are called as bacteria, but same time, virus is creating a challenging time for us because it keep on mutates, it can't keep on spreads because now it's a time of backpackers. There people are keep on moving across the globe. It is not people confined to a particular locations. Now the global travelers, they can carry a virus from India to Argentina, from Argentina to Chile, from Chile to Europe, from Europe to China. So this is how people keep. It's a global community. So problem to one country is a problem for all. Thank you.